Hello, everyone. Welcome to another one of our seminar talks by people who are developing open source tools for neuroscience. This is part of a collaboration between Open Neuroscience and the Worldwide series, where we're trying to disseminate knowledge about sciences and research via YouTube streams and online talks. Today, we have the pleasure of welcoming Dr. Phillip Kyriakakis, who will be presenting work on optogenetics, more specifically, a tool that they built in the lab, which is a simple and versatile illumination system for optogenetics. But a little bit more about Dr. Kyriakakis before we start. So he's a senior research scientist in the bioengineering department at Stanford, in the Woodside Institute for Neuroscience. He did his undergrad in biochemistry at the University of Massachusetts in Boston, where he worked also in the lab of Dr. Alexei Veraxas, in the developmental biology lab, doing biochemical experiments as well as starting to develop or contributing to the development of 5B optogenetics. He continued his education at UC San Diego at the Division of Biological Sciences, where he studied the cellular programming and metabolism, obtaining a degree with a specialization in multiscale biology. He also did postdoctoral work at the bioengineering department of UC San Diego with Todd Coleman, continuing the development of optogenetic tools and related technologies. In 2021, he moved to a senior research position at Stanford in the bioengineering department at the Woodside Institute for Neurosciences. Philip, thank you very much for agreeing to be with us today. We're really excited to hear about your work. The floor is yours. Sorry, before we start, so if anybody has any questions, please write them in the chat. Philip will kindly reply to all of them by the end of the talk, and I'll make sure that they are all curated before the end of the talk. Thank you, Philip.
Thank you. Okay, can you all see my my presentation screen?
Yeah, that's good.
All right, so I'm going to talk about some open source and inexpensive optogenetic stimulation tools, and a little bit about how we use them, so you can get a flavor of how they are applied. And I'm also going to talk about some other tools that were developed by others at the very end, so you can get an idea of what's available out there that are open source. A little bit of background about not just about me, but really about why I was motivated to develop these tools. So I did my undergrad at UMass Boston in Alexei Veraxas' lab, and I finished some projects before graduating and applying to grad school. So he let me kind of do some crazy projects where I wanted to control genes using light. And you can see here, I developed this Western blot where I activated this GFP-Kurtz gene in these cells using light back in 2008. And I think it was the first time anyone had actually controlled genes in animal cells. But a week later, I flew to San Diego and started a PhD. So I wasn't able to continue that work. I did try, but as you can see in 2008, there's only 13 papers with the word optogenetics in it. So it was very hard to find people to work on it during my PhD. And you can see now there's about 9,000 as of today. So it wasn't even a field then. Yeah, like just a little bit more of a background is like back then, what I used to control the cells were these different fluorescent tubes, the ones made for like turtles and stuff, or for reptiles where they have some far red light. And then we use these filters to filter out the red and blue light, which is what Peter Quayle did, a plant biologist who, had used this stuff, in the 90s to study plants. So that was kind of like the state of the art in 2008. Then during my PhD, just by coincidence, Todd Coleman came to UCSD, and we had this big office where he ended up right next to me, and he was making all these cool electronics with like flexible circuits and LEDs and everything. And when I saw that he was having LEDs, I was like, wow, this is really cool. And I told him about the idea of controlled genes with light, and he got really excited about it. And we thought it would be really cool to intersect our expertise and our curiosities on these topics of molecular biology and optoelectronics. So I decided to do a postdoc with Todd and further develop the 5B optogenetics. And so I'm going to focus more on the optoelectronic side of things. I'm showing this fancy flex and very small electronics here, which is what a lot of people think about when it comes to optogenetics. And people also think about like fiber optics that are surgically implanted, or microscopes or micro LEDs, and those tend to be really expensive and very specialized. But actually, what you need, really, it obviously depends on what you're doing. You don't always need the most fancy gear, like, do you need to control things spatially, or you're trying to control things in like a whole animal, for example. In our case, we mostly controlled cells in a multi-well plate, which allowed us to put a lot of different conditions into different wells, but they all have the same light illumination. And then the other thing about like choosing which system is like, are you trying to control neuroactivity, or are you trying to control genes like we were doing, or some other cellular behavior? And I'm going to talk a little bit more about how to choose them at the end, but I just wanted to point out that there's a lot of tools available, and they're not necessarily expensive or fancy. But at the time, when we started developing these, you still couldn't buy anything, and there wasn't really anything published to build off of. So Marianne and Vincent in our lab, a PhD student in undergrad, developed the software interface with the microcontroller, this Arduino, and a bunch of LEDs to control the lights. Because remember, before we were using fluorescent filters, right? 2008. So they developed this software and hardware interface where we could control the LEDs and the timing. And that was great for doing our research for developing this tool. But I knew that they were going to graduate one day, right? And so I was like, well, I'm not an engineer, so what am I going to do when they're gone? Like, what if it breaks, or I need to make some changes? And then I thought, a lot of people can't really adopt these things because there's, a ton of biologists out there that don't know engineering, and maybe they want to have this, and that's a big barrier. So I thought, what if I make like sort of a manual for myself, and maybe we can turn that into like a publication. So that led to a few different things. So first of all, we did build like a protocol to assemble it, but first we actually simplified the system. So we simplified the circuit somewhat, and we tried to really streamline it so that it would be like minimalist in a way, so that it could be more accessible and easier to write a protocol on. And then we had really step-by-step instructions on building the circuit with some illustrations and building the LEDs, and also instructions for, installing and running the software. And the system is relatively inexpensive. So, as of a few months ago, the whole thing would be about $250. You could, put more LEDs on it and things like that, and it would cost more, but that's relatively inexpensive. You do need a tablet or a computer, but it is a Windows computer, but actually you can find one on Amazon right now for about $160. And then using that, you can actually just Velcro it right onto your incubator. So we have this nice like 2 LED interface with big buttons, and you can put it on this tablet, and then you don't have to look in the incubator or open it to manipulate anything, and it has a very small footprint. So Vincent and Marianne also made this version where there's 4 LEDs, and there are smaller buttons because you need to compact it, but it's more for like a PC or laptop or something, because you can use a mouse to click on them. And then so we have these 2 different options. But we also have the option to actually just download the files from GitHub and you can make your own. So if you want to put 8 LEDs or something like that, you can do that. As I mentioned, we did do step-by-step instructions, and Lang in our lab made these really nice illustrations where, they're very detailed in showing even which pinholes all of the components go into, and instructions on the illustrations. And we even grade out like the stuff that's already assembled and highlight the next step in the assembly. So you really don't need to have any understanding of actually how it works if you can just follow the instructions step by step. Our goal was to make it so that it's really for a beginner or advanced user who can just do it quickly. And on top of that, we have pictures in the protocol where we show each step, but we also have some tips where you can, trim the wire to be a little thinner to fit through the pinhole. And then we even have some different angles of the actual picture, so you can see what it looks like and compare what you're building to the actual picture. So we're really trying to assume, nothing in terms of someone's background when they're trying to build it. And I think this worked really well with like kind of me as a biologist working with the engineers in the lab to going back and forth, because they know everything and about this, and I kind of am somewhat naive about it. So I can be like, well, that's not very clear, or yeah, I want to make sure it's really, really obvious, As I mentioned, sometimes we have a system with 4 LEDs. And again, instead of assuming that someone can take our single LED circuit and replicating it across the board, we have instructions at the end where you can build the circuit with 4 LEDs. So we really wanted to assume nothing. And in fact, we actually had a person test the build who has a psychology background, she was working in the lab and had never touched electronics before. And we were like, okay, follow these and, let us know if there's anything that, isn't obvious or something. So we had multiple testers to make it very clear. And we also have even step-by-step instructions for just soldering the wires together, crimping these, which was I actually learned doing this, which is kind of cool. So you can plug these into the Arduino microcontroller, and then these connections go into the circuit board. And then another thing we did is not only make instructions on building the LED, but we did a little bit of like sort of innovating on how to solder these LEDs. So we found that when you solder one of the wires onto here, this heat sink is really good, obviously, at transferring heat. So when you go to solder the next wire, sometimes this one comes loose. And, someone who has a good electronic setup, they might have no problem with that. But we don't know what, what tools somebody has. So we actually found that by putting these hair clips here, they're really cheap hair clips from Amazon, then they're metal, and you put them on there. And then even if this solder melts while you're soldering this other wire, it doesn't lose its place. So it made things a lot easier for us. And we also had another trick. So we're mounting the LEDs here on the solder pads using the solder paste. And what we did is we had an order where we solder the first wire, and then we place solder paste onto the pads before we solder the second wire. And then we put the LED there. And when you solder the second wire, it actually heats up the heatsink enough to solder the LED onto the heatsink at the same time. So it really makes it a lot easier. We actually tried many methods, like putting the heatsink on a hot plate to solder the LED there, but then it would melt anyway when we're soldering the wires after. So we went through a lot of iterations and found makes things a lot easier for people to do. And then we also encased the LEDs in epoxy so that the wires are nicely attached. They don't come off easily. And it also is better for coating all the wires and everything for if you're putting it inside of an incubator or something like that, different types of environments. And we've actually had them in our incubator for weeks and months at a time. Sometimes I would have them all blinking and stuff just to kind of abuse them and see, if they break down, like how they break down. And we haven't had any issues with them. So it really works great to have these things coated and they're nice and robust. And putting it on the heat sink, we were also then able to just put this command strip, which is like a Velcro on the back, and it's really strong and it can go on pretty much any surface. So you can put it inside of an incubator. In this case, we mounted it inside the top of a black box. So you can put your cells in this black box and stimulate them from the top. And of course, you can put more than one in the box and move it around. So it's very modular. You can put it inside the entire incubator on the wall or on the ceiling. Molly Allen, a PhD student in Peter Wang's lab, took one of our systems and did some adaptation of it. So she put it in this white box at the bottom of the box and put this privacy film on the top of the box, and then put the LED there and turned it on and flipped it over on top of the cells. And what that allowed is for the light to diffuse very evenly over the cells. And in her publication, she measured the distribution of the light and got it to within a certain range that worked for her. And yeah, it just goes to show that, if you're creative, you can use these in different ways. And, it doesn't cost a lot of money to have like a nice optogenetic system where you can publish a paper on it. And also the system really allows for a lot of different wavelengths. So you can do all sorts of different optogenetics. So if you want to do like a channel rhodopsin activation with blue light, you can use these LEDs. We also found that these heat sinks from a different manufacturer from the very popular ones here have a lot of wavelengths in the near IR region, which is good for a lot of different types of optogenetics. They have a lot in the visible spectrum as well, but these are kind of more popular, so I put them on this chart here. And you can actually buy some of these already pre-mounted on the heatsink, so it's a little more expensive, but you can't buy these pre-mounted. So I think the instructions on how to mount them is pretty useful, so you can kind of put any LEDs you want on there. And here's a picture of one of the assembled boxes. So here we have a 4 LED system, and we control the intensity manually with these knobs, and we control the voltage instead of the current, which I know a lot of people prefer. But the reason we do that is that by controlling the voltage, we can use these different LEDs, which have some different specifications for voltage, right? So it allows us that flexibility. And as I said, we've kept them in the incubator running and blinking for weeks at a time without any issues. So this really works great. And we learned that if we label these wires, even though this looks like a lot of stuff, like it's actually pretty easy to snap together because you just match the wires coming out of the board to the wires, in this case, the potentiometers or the Arduino connections. So it's fairly easy to assemble as well. We also wanted to make sure that the software instructions were easy. So my students would zoom through this and be like, oh, this is how you do it. And then I'm like, a month later, like, what? I don't remember. I want to program one. So I did this and screenshotted and made nice arrows to show exactly where you click to program this Arduino. I also did the same thing for installing the software, and I have instructions for running the software interface as well. In addition, we put the operating instructions on the interface itself so that, when you're using it, if you haven't used it in a while, you can just read this really quick. And so here you just control the connection to the Arduino. And then here you have your LED control. So you can program it to blink. So you can have it on for 1 minute, off for 1 minute, or you can have it just constantly on, or whatever, program you want. And then you can do a start delay. So you can put it in your incubator and wait 24 hours before the LEDs turn on, and you can control the runtime. And then it will also tell you here if the run is complete. So if something went wrong or whatever, this will say not yet. And then it'll say complete when it's done, and it'll change color. So yeah, I really try to make this very user-friendly. And as I said, we have the other version, which is basically the same thing, but more LEDs. And so it can really do some, a lot of different experiments and optimization. And in fact, most of our experiments were done with just 2 LEDs. And I'll show you next the kind of experiments that we did do with this. and how we were able to use it to characterize some optogenetic tool. I need to give a little background on this tool though, because it's a little different from having a neuron fire when you shine light. So rather than a neuron firing when the light is on and then going to the resting state when the light is off, this system is bistable. So it turns on with red light and shuts off with far red light. So what happens is this Phi-B, this plant protein, and this chromophore will absorb the red light, and then it will dimerize or bind to PIP3. And Peter Quayle was studying this interaction when he developed this light switchable promoter system in 2002. And the way that worked is this PIP3 is fused to an activation domain. And so when you shine light, these come together, and the activation domain is recruited to the DNA and near a promoter and activates your favorite gene. So when you shine red light, this interaction becomes very stable, and the gene will stay on for a long time, and I'll get into how long in a moment. But it'll do that until you shine far red light, and then it will shut off. And while he showed that in yeast, we wanted to see that that was working in our mammalian or animal cell system. So we did that by a relatively simple experiment. So we shined light. We had the LED box with just a red LED in one, and then a red and far red in another box, and another box with just far red. So we shined red light for 12 hours in these two boxes, and then shut off the red LED. And this one stayed in the dark, and this one stayed in far red. And you can see the one that stayed in the dark still had some gene activation compared to the one that was in far red. And obviously the one that was always in far red had very, very minimal activation. And then the one that was in constant red light had the most gene activation. So it really shows us that this is reversible. It's expected, but it was good to confirm in mammalian cells with our proteins. We were also able to look at the light sensitivity, and we actually characterized it in several different ways, because as I'll show you in panel D, that the results weren't totally clear, and we realized that the way that people report light sensitivity of a lot of the systems is different. So you'll see a review article, and they'll have a list of optogenetic tools, and it'll say, here's the sensitivity, and maybe they'll give a range too. But as I'll show you, that's a little bit misleading because it depends on the context. So what we did here is we had a 24-hour pulse of light, where before we ended up measuring the samples for the gene activation. And you can see here with these first three samples that where we had lowering amounts of light, and we do this in micromoles per meter squared per second. Those are the units of photons, but I'll just call it micromoles for now. There's not really a big difference with these three illumination intensities. But then as you do it 1/10 the amount of this one, you have a significant difference statistically. Now we're like, okay, but it looks weird. There's a little trend going down. What's going on? And then we're like, what about 24 hours? What if we shine less light? What's the sensitivity? And by less light, I mean not intensity, but time. So we did this experiment where we said, okay, we'll just do a 1-minute pulse, right? So what's the sensitivity with these different and same exact intensities as this last experiment? Well, there's two big differences. One, look at the scale. Okay, so this is 30-fold gene activation compared to 200. And then when you're doing these lower amounts, you barely get any gene activation. So it really tells us, okay, so what if I publish a paper and then a review article, and then I say it's this sensitive, but I'm reporting something from a 24-hour pulse compared to a 1-minute pulse, right? So it really does matter, how long you're activating the system. So just got to think about that when you're looking at the reviews and picking a system. Make sure you go and look at some of the different experiments that were done to determine that sensitivity. So given this, we were, and given the reversibility I showed before, we were motivated to further analyze the sensitivity. So we looked at the photon titration, we call it, where we have two different amounts of light here. So this is light intensity, where it's the same conditions as this experiment here. And then we also have experiments where we pulse the light using our LED system. So you can note here that this is 1 minute of light on, 9 minutes of darkness, pulsing for 24 hours. And that's the same amount of light as this condition here, where it's 1/10 the intensity, right? Because here it's 1/10 based on time. And we're getting kind of a similar result based on constant light and pulsing light. When you go lower than that, it's lower gene expression. But when you're at this level or above, it's the same as constant light. And the amount of photons is the same. So after you get below that point, whether it's pulsing or continuous, you're getting less gene activity. So it's really reassuring to us, no matter how we did the experiment, the way we measure the light sensitivity is consistent. But then we were also like, okay, we wanted to get to the point where it starts getting less and less gene activity. And we were kind of shocked that even though you only shine light for 1 minute and then it's off for 29 minutes, that it's this similar to when it's constantly on. We think of like optogenetics to be really, you shine it and it turns on and off, and even the bistable system to be on for several minutes is, we thought that was kind of surprising. So we did another experiment where we had, this is a different version of our LED system that had more, 8 LEDs, but we did pulsing again with 29-minute intervals. But then we also did every hour and 4 hours, 8 hours, 12 hours. And note that the 12-hour one and the 8-hour one only have 2 pulses and 3 pulses. So very little light pulses. And we did it at this higher intensity of light. So we wanted each pulse to be saturating in terms of the light intensity. So we knew it was saturating from this experiment where you have 0.1 and 1, and you're getting the same amount of gene activity. So we picked 1 here. Each pulse should totally activate the system. And again, to our surprise, even when we pulsed every 8 hours, we're getting about the same level of gene activity as the shorter pulses. So, and even at 12 hours, which is only 2 pulses, we're still getting like 100-fold gene activation. So the system is very stable once you activate it. And so it could be very useful where you only need to shine light like 3 times a day for something, right? But also what we did then is we calculated how much light it got in total. So the total flux, so that's in micromoles per meter squared total, not over time. And so you can see that this is only 3 minutes of light, right? Total, so it's very little light. And so that's 180 micromoles per meter squared. These experiments here with the blue star are the same amount of light. So that's 864 micromoles per meter squared. So whether it's by dimming the light or by pulsing it, it's the same amount of photons, right? And then when we did this experiment, we found that the 8 hours was, not significantly different from faster pulses. So we liked also that these agreed. So you see here that these lower amounts of light were about the same as the higher amounts of light, which is in the same range as these experiments. So we did 3 different types of experiments, and they all had pretty much, we're all in agreement in terms of the light sensitivity. So by using this system, we were really able to thoroughly characterize the optogenetic system in terms of its reversibility, the sensitivity, the bistability. Yeah, most of it was done with 2 LEDs. This one was done with more. But you can see what you can do with a relatively simple LED system. And I wanted to mention another caveat that, or something I got a lot of questions about from people who want to do optogenetics. One is how much gene activation am I going to get? And another one, which I think are people who are really like skeptical, or is like, how much leakiness am I going to get? And then having done this, like I optimized some stuff before we did the experiments for our paper, and I had experience with it, so I kind of explain it. But then I keep getting these questions, so I thought for this Jove publication, why don't I do the experiments to really quantitate it and show it? So basically, this is intuitive. So if you have a DNA where you get transcription of a gene that is turned on with light, but off with in darkness, right? There can still be some level of transcription in the darkness, right? Because biology has some inherent noise, right? And if you have more of these in the cell, there's going to be more noise, right? But I wanted to quantitate it. So what I did is I put more and more of this reporter DNA in the cell, and with some filler DNA so that they all get the same amount of DNA total. And then you can see here, these two lines is I put higher concentration of this reporter DNA. And I put it in red and far red light, but it didn't actually have the optogenetic switch. I just did that as a control to make sure the light wasn't doing anything, right? And you can see here that there's more gene expression, the more DNA I add, even though there's no optogenetic switch there, right? So that's kind of like measuring the baseline or the basal level of noise of the system. And then when I add the light switch there, but I have far red light, which is our off switch, right? So that the gene system should be off. You actually get more noise, but it also increases when you add more DNA, which makes sense. But as I said, like people ask about it, and I wanted to have something to show like data-wise, rather than just say, well, it depends on how much DNA you put there, you know. But then the other question is, how much activation do you get? Well, I put the same data, this far red data, which is our off switch, on a different scale with the red light. So it looks very different, right? It looks like there's almost nothing there. And then I put the data from when you activate the gene system with the red light here. And you can see here that there is activation compared to far red light, but your activation level actually goes up too when you have more DNA. So the take-home message is you really want to think about what you're doing in terms of what your protein that you're expressing is. You have to think about, like, how much of it do I need for measuring some type of biological activity? Are you expressing something like an enzyme that recombines DNA irreversibly? If there's one molecule of that in the cell, well, then you might get recombination in the dark, right? But if you want to express something like a fluorescent protein, then you may just filter out some of the basal expression with your camera settings, and then but you want higher levels so that you can get a really nice bright picture. So you need to think about what the experiment is you want to do with it, and then kind of like optimize the amount of DNA and components of your optogenetic system. And this is actually true not just for optogenetics, but like any chemical inducing system, and even not even controlling genes with light, but controlling other processes in the cell. They're all going to have these same types of things that you have to consider. And then another thing people might wonder is, okay, but does the fold activation change, or the signal to noise ratio, when you have more DNA? And I mean, it doesn't really seem to be impacted. You get more protein expression with more DNA, but the signal to noise ratio is about the same. So the take-home message of that is the amount of expression you get with an inducible system is really relative. It's modulated by copy number of your DNA and probably the expression levels of your optogenetic tool as well. And so design the system wisely and be prepared to set it up so that you can try different things so that it works well for your application. So another thing we wanted to optimize as well, or characterize, I guess, is how far into the tissue can you activate the genes or your optogenetic switch. So the first thing I said is, how sensitive is the light switch? Well, your light switch is also as sensitive as it gets into your tissue. So if you have blue light, even if you have a sensitive switch, if the light isn't reaching your target, your light sensitive molecule, you're not going to get any activation. You really have to know how far it's going to penetrate, and by that I mean like what intensity of light reaches the target, right? And so that's where red light is really beneficial, because you can see here, some person's hand, they don't absorb beyond like 650. It becomes very translucent. And this may depend on your hand, your skin color, the thickness, of course, but you can see here in my friend's hand that you can see through her hand pretty well. If you do this with other wavelengths, you don't really see much go through. So it's really beneficial to use wavelengths in this 650 to 900 region. We were writing a grant on studying hearing using optogenetics. And so we thought we want to measure the amount of light that reaches the ear. And so we had this isometric optical probe that measures light from all the surrounding angles. And we put it in the temporal bone of the ear and had a 15-watt LED lamp above the mouse. And this lamp was placed right at the height of a typical mouse cage. So this is another more than open source. It's just a lamp you can buy on Amazon. You don't need anything fancy here, right? Just a 15-watt red LED lamp. And we measure the light in the ear, and we're getting 30,000 nanowatts per square centimeter. So, very conservatively, you need 180 nanowatts per square centimeter to totally saturate our system. So we could easily activate genes in the mice ear using just a lamp above the cage. We also placed the probe actually under the head, shielded with foil, and we placed the LED at different distances above the mouse. And even at the 20 centimeter distance, we're getting more than twice as much light as we need to activate our system. I should mention, this is done with Leo Andrade and Uri Manor at Salk Institute. So this is really cool because this is a really big mouse actually, and it had black fur. So in this setup, the lamp is actually putting light through the fur, the skin, the skull, the brain, more bone, more skin, and more black fur before it hits the probe. And it's enough to activate the system that way. So we think just by having a lamp over the cage, you can actually control the genes pretty much anywhere. And you could have rather than putting fiber optics and wires and everything in the mouse, you can actually just have these lamps above and control whatever number of mice are in your cage. And there you can easily do multiple cages. So imagine the throughput compared to doing some surgery to put your LED there. So this is a really nice way to do it. And it was also recently done, actually this was published last month, where someone used a different red light system. It's actually a similar phytochrome, and they put the encapsulated light sensitive cells, it was right under the skin, but they put them in a mouse, rat, and in rabbit, and shined a lamp over them and was able to control the genes in these animals. So, it's really low tech in a good way. You don't need to have any expertise, right? You just need to have a light meter to make sure you're, illuminating them enough, and have a cage that is appropriate for that. And then another thing that we did with these lamps is, because our system shuts off with far red light, we actually had the far red light room using these lamps. We shut off all the lights and would do our cell culture manipulations in the cell culture hood with our lamp pointing towards our sample, so that we're not activating it while we're changing media and things like that. And then another similar low-tech sort of way of doing this is Christopher Voigt's lab, when he was at UCSF, was using a confocal to control these cells, so a laser spot, a very small laser spot. But he actually had far red illuminating here by taking the bright field lamp and filtering out all the light except the far red light and shining it everywhere, and then having a brighter laser spot to control the cell. So it was shutting everything off around the cell, but then there was so much more red light here that it still activated the system, and they were able on a microscope level control the cell shape using the light. So those are that's another clever way to use off the shelf sort of optics. At least this is a confocal microscope, but the part that's the bright field is on every microscope basically, right? So I also am going to talk about some other systems that are available that I think could be useful depending on what you're doing. So here's a system that's actually very similar to ours in that It uses the same LED as one of the ones I showed, and same heat sink. However, they use some lenses to make it shine more of a distance. So they don't have all the instructions on how to make the LED and everything. So you can always use, that part of our paper to do that. And they did put a heat sink on it because they're controlling channel rhodopsin with blue light in worms. And that requires literally a million times more light than our system. So it's generating heat, and you really need this big heat sink. But what's nice about it is that you can do it at a distance. So you can do some different things where you can image the worms here and have the light come at a different angle. And this is all open source as well. And so that's another option depending on what you're doing. I know this is an open neuro talk. A lot of people want to use channel rhodopsin. So you could potentially build something like this to control channel rhodopsin and use like a hybrid of this in our publication as well. If you want to do something in multi-well plates, sort of like we did, but instead of illuminating like these 4 wells all with one LED, if you wanted to control individual wells, these 2 publications do that. This one actually controls the cells with multiple wavelengths in one well, and they have a good range of different wavelengths. So these are a little more sophisticated to build, maybe a lot more and more expensive, but they have different uses, right? So sometimes you may want to do a bunch of different conditions in parallel. These are all open source as well. So these are options you can use. This is kind of like the next level up. So this one was from Lucas Bugaj and Wendell Lim. They developed a 96 well version and actually a 384 well version where they actually have 2 LEDs per well. So this is really cool. A lot of people may not need this, but you could do like a lot of interesting experiments with all of that. It's definitely more expensive and requires more expertise to build, but it's open source as well. So they do have instructions. This is a really, really cool system. So you don't really need to buy one. It's definitely going to be cheaper than buying something. And I don't think you can buy one that has 96 or 384, at least not last time I've checked. So, my take-home message is that you don't always need sophisticated, electronics and things like that to do optogenetics. It really depends on what you're doing, and it doesn't have to be expensive. We developed a very simple system that has a lot of detailed instructions, and it's a Jove, so we were going to make a video, right? But because of COVID, we were not able to. And actually, our new lab is still under construction. It's almost done. So hopefully we'll have the video recorded and up soon. It'll add another layer of detail on building these things. And using our system, you can control many types of optogenetic tools. So as I showed, Molly Allen used it to control a light-controlled enzyme, Cree. And then another person in Peter Wang's lab used it to control T cells for a model of cancer therapy. And we used it to control a reversible red and far red light system. So you can really use it for a lot of different things. And also the other group used a very similar system for neural control with worms, right? You can use it to optimize and characterize your tools. Usually that's when you need the most different conditions and stuff. Once you have it optimized, you tend to use just one LED for stimulating or something. But you can, with 4 LEDs or with multiple systems, you can really characterize a lot of these things in detail. And also another thing is to remember to choose your system based on the biology. So, your signal to noise matters, what you're expressing when you're shining light matters, what gene it is. But even if it's not a gene control thing, if it's some other optogenetic tool that does something else, you really need to consider the biology before you really decide what system is best. It's not just going to be your wavelength or what electronics you have. You don't want to be stuck where you're doing something and you realize it's just not enough light, or I'm not getting enough expression. So set it up so that you can try different things very easily. And as I showed at the end, there's also other systems, not just ours. Sometimes you can even just use a lamp, right? There's going to be more too. So I hope that more people adopt the optogenetics and don't think that, like, it's something that, has a barrier, a cost barrier or a technical barrier of, okay, I don't have electronics background. So maybe I shouldn't do these optogenetic stuff. You really can get different things that are open source depending on what you need. So with that, I wanted to thank the contributors. So Marianne developed a lot of this, the electronic software, and did a lot of the optogenetic experiments with me with the cells. And then of course, Todd Coleman, who was my postdoc advisor, and we are still working together now. And hi, Todd, I see you. Gentry and Lara are our collaborators, doing some of the cloning initially, and Nicole, Vivian, and Walter did a lot of the cloning and the cell experiments. And Vincent worked a lot with Marianne on developing the hardware, software, and also did some of the cell experiments. So I mentioned their names here too. And actually, my dad helped simplify this circuit. And Lulu, actually, she's the one I mentioned with a psychology background initially. She helped kind of test building the system to see, like, okay, I don't have any background in electronics. Like, can I follow this, And I really want to thank Lang for these really nice illustrations. I asked her to help with that, and I was blown away when she showed me even the drafts. I was like, okay, that's way better than I expected. And then Leo and Uri for helping with the measurements for the mice and putting the lamp, and of course our funders too.
Philip,
I'll take any questions.
Sorry. Yeah, thank you. It was really great. We do have questions. The first one is from Jag. So the question is, for how long would one need to stimulate to get enough protein expression to possibly alter the behavior of an animal, assuming enough neurons can be stimulated?
So that's protein expression. I guess there's two things in there. One is how much protein would you need to express with the light, and the other one is stimulating neurons. Okay, so I guess it would depend on what you're using, what protein you're expressing. For example, at one point I had the idea of expressing a dominant negative version of a protein that would make the natural occurring ones dysfunctional. And that actually would change the whole gene expression profile of the neuron. And that would require a lot less than if I'm trying to overexpress something that is already highly abundant in the cell, right? So it really depends on your application. Proteins, their half-lives and their abundance span like many orders of magnitude. And so I can't say that for any specific protein. But I think if you want one that is probably in the middle is my best guess, something that is not functional with 2 or 3 copies in the cell, and not something that's like actin where there's like so much expression already. Not that actin would necessarily change anything, but if it's super high abundant already, it's going to be hard to make it more abundant, right? So it really depends on the protein that you're interested in.
Okay, thanks. So he did follow up with, assuming protein expression can change the activity of a neuron, but I think you already covered that, right? So I think it was just to clarify. Then we have, a question from Heather, which is basically, what age of mice did you use for your in vivo tests? And she says, continues to say, we have a model of juvenile epilepsy where the pups die before weaning age and so are never large or healthy enough for surgery. Being able to use optogenetics without fiber optics could be fantastic.
Yeah, off the top of my head, I don't remember the age. I'm not an expert in mice. That's Leo and Uri's kind of background, but I think it was about 2 years old. It was a very old mouse. It was very large. actually our goal was to probably somewhat similar to yours in some senses. We actually wanted to look at the very early stages of development. So we were wanting to shine light in the pregnant mouse to control the genes, before they're born, before the mice are born, and after, but also before. So that was kind of our goal, but we had like an older mouse to test it on because it was already old and not useful for other people, and also it was very large. So we thought it was like the best test case to see how well the light penetrates.
Okay, cool. So I actually have a question, if you don't mind. You showed these leak experiments, right, where you had the protein being expressed without the optogenetic system, right. But then you also showed that by illuminating the system with the light that turns everything off, you also get expression, right? Did I understand that correctly?
Yes.
So my question is, could this be because the LED that turns the whole thing off, the spectra of it is actually quite broad and a little bit like spans the actual side of the system, like the actual side of the spectrum that actually activates the system?
Yeah, that's a good question. I think that that's a tiny part of it potentially, but it's actually more than one factor, I think. So one of them is just that proteins, whether they interact or not is not binary. So it's really about binding rate on and off. So when they're in the off state, they can still bind for just less, right? The other thing too is that in the experiment without that, so I had let me go back to it actually. Yeah, so in these two conditions, I had just the reporter DNA. So there was no optogenetic switch. And then here I include the reporter DNA. So again, like here you just have the DNA, no optogenetic switch, right? And then when you include these proteins, they're binding on and off at a very low rate when they're in far red light, but they're more bound in the red light. So even though this is the off state, there is going to be some level of transcription going on. But it's pretty low if you consider the difference between the far red light and the red light. And that's reflected in our experiments here. You're getting like 300 or 250 fold activation. So you're getting 250 fold difference between these conditions. Oh yeah, another factor, by the way, is it's kind of what you were saying in a different way. So I think the LEDs are far enough apart because the 650 nanometers and 730 is pretty far apart for LEDs. They have a fairly narrow emission spectrum, but actually what's not narrow is the absorbance spectrum of the chromophore. So what happens with the chromophore is you have a red light absorbing state, and then it isomerizes when it absorbs a red photon, and now it has a far red absorbance spectrum. And there is a little bit of overlap between the two. So the red light absorbing species or isomer absorbs a little bit of far red light as well. So it does tend to activate, but then it also deactivates it. So it's a little complex. It's not just the optics, it's the biology too.
See, okay, cool. So there are no more questions coming through the chat. I would like to say if people do have questions, I guess this is now the moment to have them. So please do write them in the chat if you have them. I would like, in the meantime, as we give like people a couple of seconds to maybe ask something, I would like to say thank you again. This was really great. I really, really like the approach of assuming nothing about the knowledge that people have and we're producing this, because this is what methods in science should be all about, right? People being able to replicate it no matter what. So I really appreciate it. I think this is a very good example that we should be following for our documentation things as well. I don't see any more questions in the chat. And so if you would not like to add anything else, I guess we can complete this session and say again, thank you very much for you and the people that attended today and asked questions and just watched the talk. Thank you very much.
Thank you for the opportunity to come. I'm happy to share my science with everybody.
Absolutely. Okay, we are officially offline.